Hello everyone, welcome to Introduction to Virology. My name is Kaylee Hash. I'm a senior at Purdue University obtaining my bachelor's degree in animal sciences with a concentration in pre-veterinary medicine and a minor in biological sciences. My ultimate aspiration is to earn my doctorate of veterinary medicine so that I can unite my compassion for animals with my passion and interest in biomedical science. One of the scientific disciplines that I find most intriguing to learn about is microbiology. Within the field of microbiology, there are many subtopics in which I find particularly fascinating, one of which is virology. Virology is the branch of science that deals with the study of viruses. The majority of this presentation will focus on providing a comprehensive introduction to fundamental topics in virology. Learning objectives will include identifying structures of viral anatomy, differentiating viruses based on their classification categories, understanding the fundamental mechanisms of viral infection and replication, and knowing main modes of viral pathogenesis. In addition, the latter portion of this presentation will feature a focus on the pathogenesis of novel coronavirus SARS-CoV-2 or COVID-19. Concepts, principles, and terms discussed earlier in the presentation will be applied with the goal of understanding the origination, structure, pathogenesis, and epidemiological significance of the novel COVID-19 virus. Before covering the specific terms and concepts related to viruses, let's review some fundamental biological macromolecules that will be crucial for understanding viral structure, replication, and infection. The main biomolecules of importance in this discussion on virology are nucleic acids, proteins, and lipids. Nucleic acids are polymers composed of individual monomers called nucleotides. Nucleotides are molecules that contain a pentose sugar, a nitrogenous base, which can be adenine, thymine, cytosine, guanine, or uracil, and a phosphate group. As you can see in the diagram to the right, in a polynucleotide chain, the sugar and phosphate groups interact to form a helical sugar-phosphate backbone, leaving the nitrogenous bases to protrude from the backbone. The two main nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Nucleic acids have a significant role in viral anatomy because they are the constituents of the viral genome. Every virus contains some form of DNA or RNA as its genetic material. Nitrogenous bases can hydrogen bond with other complementary nitrogenous bases. If the hydrogen bonds form intermolecularly, or between two nucleic acid strands, then the nucleic acid is referred to as double-stranded. If the hydrogen bonds form intramolecularly, meaning within the same nucleic acid strand, then the nucleic acid is referred to as single-stranded. An example of a single-stranded nucleic acid is illustrated in the far right of this diagram. DNA is most often double-stranded, while RNA is mainly single-stranded. However, both DNA and RNA have the capability to be single or double-stranded. In this regard, nucleic acids serve as a category for viral classification. Viruses can be categorized based on their replication strategy, and the mechanism for viral replication is determined by whether the viral genome contains RNA or DNA, and whether that RNA or DNA is single or double-stranded. Another important macromolecular component of viruses are proteins. Proteins are polypeptides formed from sequences of amino acids. Let's recall that every individual protein has primary, secondary, and tertiary structure, and sometimes quaternary structure if the protein is composed of several subunits. The overall three-dimensional structure of a polypeptide is called its tertiary structure. It is a result of protein folding that occurs due to interactions between the unique R groups of the amino acids that make up that particular protein. The tertiary structure of viral surface proteins has an essential function in viral infection because the 3D structure of proteins on the surface of a virus will govern that virus's ability to bind to a specific host cell receptor. Binding specificity allows for a virus to gain entry into the host cell. Viruses must invade a host cell with protein synthesis capabilities as the virus does not possess the cellular machinery to produce proteins that are essential for further propagation on its own. The third biological macromolecule we will review is the phospholipid. Phospholipids are major constituents of biological membranes due to their unique structure. They consist of a hydrophilic, which means water-loving, head, and a hydrophobic, which means water-fearing, tail. In aqueous environments, the hydrophilic heads interact with the water, whereas the tail regions are excluded by the water and congregate together. Moreover, these two structural components allow phospholipids to form what is known as a lipid bilayer, where hydrophobic tails face inward towards each other and hydrophilic heads face outward, interacting with the aqueous intracellular and extracellular environment. Some viruses have the ability to surround or envelope themselves in a portion of the cell membrane of their host. In this way, the virus gains an outer lipid bilayer known as a viral envelope, which we will soon discuss the structure and function of in more depth. As defined in the textbook of medical microbiology, published by the University of Texas Medical Branch, viruses are obligate intracellular parasites, which contain either an RNA or DNA genome surrounded by a protective protein coat. Viruses are obligate intracellular parasites because they are non-living. Thus, they are completely dependent upon host cells to supply the complex metabolic and biosynthetic machinery that is necessary for their reproduction. For a reference diagram on viral anatomy, we will examine the structure of human immunodeficiency virus, or HIV. According to a research paper in the Italian Journal of Biochemistry and Biophysics published in 2020, HIV is one of the best characterized enveloped viruses with the identity, roles, and structure of the key molecules required for infection well established. HIV, as stated before, is an enveloped spherical virus. The viral envelope, shown here in orange, is composed of phospholipids, and it is derived from portions of the host cell's membrane. It is embedded with some host cell proteins, as well as transmembrane glycoproteins that are coded for by the viral genome. These viral glycoproteins, otherwise known as envelope or spike proteins, play a key role in attachment and penetration of viruses into the cell because they engage cell surface receptors and mediate membrane fusion, which makes it easier for the virus to infect host cells. Enveloped viruses such as HIV also contain matrix proteins, which are illustrated in this diagram as yellow spheres. Viral matrix proteins are structural proteins that associate with the inner surface of the viral envelope. They play a crucial role in virus assembly, and they serve to connect the capsid to the viral glycoproteins inserted in the lipid bilayer. Due to their function of connecting internal components of the virus to the outer transmembrane proteins, matrix proteins are involved in viral budding from host cells. Moving interior in the diagram, the viral capsid is the protective protein coat, shown here in turquoise, that surrounds the viral genome, which is composed of nucleic acid. It serves to prevent the viral genome from being degraded by nucleases. Additionally, during infection, glycoproteins within the capsid attach the virus to specific receptors exposed on the prospective host cell. Together, the capsid and nucleic acid of the virus form a complex that is referred to as the nucleocapsid. Some viruses also contain virus-associated enzymes. As stated in a research paper published in the Journal of Infectious Disease in 2017, virus families such as negative strand RNA viruses and retroviruses contain enzymes required for early steps in virus replication. Reverse transcriptase, as seen in this diagram, is required by some RNA viruses in order to produce the mRNA transcript, which is essential for proper assembly of new virus particles. After covering the key structural components of viral anatomy, we can begin to understand the viral classification categories that arise from the structural variations between viruses. Variation in shape of the capsid serves as one basis for the classification of viruses. The capsid is composed of repeating protein subunits called capsomeres. Generally, capsids have either helical or icosahedral symmetry. Helical nucleocapsids, as shown in the top diagram, consist of an array of capsomeres wrapped around a helical filament of nucleic acid. Icosahedral capsid symmetry, as shown in the middle diagram, gives viruses a spherical appearance at low magnification, but the protein subunits are actually arranged in a regular geometrical pattern. Some viral capsids, however, are neither helical nor icosahedral. They assume a distinct structure known as complex, where they possess an icosahedral capsid head bound to a helical filamentous tail. Many bacteriophages, which are viruses that can infect bacteria, are complex-shaped. This complex virus structure is beneficial for non-enveloped viruses because it allows them to insert their genetic material into a host cell without having to cross the plasma membrane. Another basis for viral classification is the presence or absence of the lipid envelope. Viruses without a lipid envelope are referred to as naked, and they cannot enter their host cells via membrane fusion. As stated in an article published by University of California Davis Libraries in 2020, the viral envelope can give a virus some advantages over other capsid-only viruses. For example, they have better protection from the host's immune system, enzymes, and certain chemicals. The proteins in the envelope can include glycoproteins, which act as receptor molecules. These receptor molecules allow host cells to recognize and bind the virus, which may result in easier uptake of the virus into the cell. Most enveloped viruses depend on their envelopes to infect cells. However, because the envelope contains lipids, it makes the virus more susceptible to inactivation by environmental agents such as detergents that disrupt lipids. Another category of viral classification is the Baltimore classification system, where viruses are grouped based on their chemical composition of their genome, as well as their process for mRNA production. In the Baltimore scheme, as shown to the right, viruses are sorted into groups 1 through 7 based on their genomic nucleic acid makeup. Based on the genomic nucleic acid that a virus begins with, the steps that it undergoes for replication and protein synthesis will vary. Some viruses begin with double-stranded nucleic acid, whereas other viruses begin with single-stranded nucleic acid. The genomic RNA strand of single-stranded RNA viruses can be referred to as sense, denoted by a plus sign, or antisense, denoted by a minus sign. The sense strand is the coding mRNA that is utilized during translation for protein synthesis. The antisense strand is complementary to the sense strand. As illustrated by group 4 viruses that already begin with sense single-stranded RNA, an antisense strand must first be synthesized by a viral RNA polymerase in order to produce more sense mRNA. This is because in order to produce more sense strand mRNA from the copy present in the genome, the viral RNA polymerase must first utilize an antisense template. With a basic understanding of viral structure and classification, one can begin to appreciate the incredible diversity of viral morphology. Due to variations in capsid structure and symmetry, presence or absence of a lipid envelope, and differences in nucleic acid type, viral morphology has many branches. Here are just 4 different types of viruses that are common causes of human and animal diseases, pictured along with their corresponding electron micrographs. Adenovirus is a culprit of the common cold in humans, and strains of canine adenovirus can cause infectious hepatitis in dogs. It is an icosahedral DNA virus without an envelope. Fibers extend from the 12 points of the icosahedral capsid, and DNA forms a ribbon-like molecule. The electron micrograph of adenovirus can be seen in the top left corner. In the top right corner, HIV is the etiological or disease-causing agent of acquired immunodeficiency syndrome, or AIDS. Its glycoprotein molecules protrude through the lipid membrane, and the icosahedral capsid encloses a nucleocapsid in which the RNA is contained. Influenza virus, shown in the bottom left corner, is commonly known as the flu. It is an enveloped RNA virus containing a nucleocapsid of helical symmetry. Enveloped spike proteins protrude from the lipid bilayer of the influenza virus. Shown in the bottom right corner The rabies virus is a virus capable of infecting nerve cells in humans and animals. It has a helical RNA nucleocapsid with a bullet-shaped lipoprotein envelope in which approximately 200 glycoprotein envelope spike proteins are embedded. As illustrated by even this small sampling of 4 common viruses, viral structures all contain at least the 2 main elements of the nucleic acid and the capsid protein coat, regardless of variation in size and shape. Structural variation is what allows viruses effectively to invade their targeted host cells and replicate, which we will discuss next. Viral replication can be outlined in 6 steps: attachment, penetration, uncoating, replication, assembly, and release. To enter new host cells, viruses must penetrate the barrier presented by the plasma membrane of the target cell. Many enveloped viruses are able to enter their host cell by membrane fusion, where the viral envelope fuses directly with the cell membrane. Plant and animal viruses that do not have an envelope can enter through receptor-mediated endocytosis, in which the host cell plasma membrane surrounds and engulfs the entire virus after the virus binds to specific receptors present on the host cell membrane. The diagram to the right depicts a virus entering its host cell via endocytosis. For bacterial viruses, or bacteriophages, the nucleic acid enters the host cell naked, leaving the capsid outside the cell. During attachment and entry, the bacteriophage attaches itself to the host cell and injects its genetic material into it. Prior to covering the next steps of viral infection, another brief cell biology review is in order. The central dogma is a foundational principle that outlines the flow of genetic information within a biological system. It differentiates the processes of replication, transcription, and translation. Since the process of viral replication depends on the type of nucleic acid originally present in the viral genome, as we discussed in the overview of the Baltimore classification, it is important to be able to distinguish the different replicative processes from one another. This diagram is a useful tool for understanding the key function of each replicative process, as well as the key enzymes associated with each replicative process. Sometimes viruses contain these enzymes in their capsid in order to aid with replication. Replication, in terms of the central dogma, specifically refers to the production of DNA from a DNA template or RNA from an RNA template. Transcription refers to the production of mRNA from a DNA template, and reverse transcription refers to the production of DNA from an mRNA template. Translation refers to the production of protein from an mRNA template. Now that we've reviewed the processes used by viruses to replicate their genome and produce proteins, we can proceed with the next steps of viral replication. After attachment and entry into the cell, the viral capsid is degraded, and the viral nucleic acid is released in a process called uncoating. During uncoating, the viral DNA or RNA incorporates itself into the host cell's genetic material and induces it to replicate the viral genome. The location and mechanism of replication depends on the type of viral nucleic acid. DNA replication usually happens in the nucleus, while RNA replication typically occurs in the cytoplasm. However, there are exceptions to this generalization. Regardless of where the replicative process occurs, there are 2 main objectives of viral replication. One objective is to replicate the nucleic acid of the viral genome, and the other objective is to produce sense, positive strand mRNA that can be translated into protein. The viral RNA of positive strand RNA viruses acts directly as mRNA for protein synthesis. Once proteins have been produced from the positive strand mRNA and nucleic acid has been replicated, the viral nucleic acid and protein can then assemble into a new nucleocapsid. During release, the newly created viruses are released from the host cell, either by exocytosis or by budding off through the cell membrane. Let's transition on to understanding how viruses are able to cause disease in their hosts that they infect. Viral pathogenesis is the process by which a viral infection spreads throughout the host and leads to disease in a target organ or tissue. Disease severity can range from subclinical meaning that the disease is inapparent and does not cause symptoms in the host, to mild, severe, or even lethal. Pathogenesis occurs in progressive stages: implantation, local multiplication, local spread, and dissemination throughout the body. Viruses are carried to the body by all possible routes: air, food, bites, and any contaminated object. As part of their entry and infection strategy, viruses interact with specific receptor molecules expressed on the surface of target host cells. Most viruses have an affinity for specific tissues, which is a phenomenon known as tissue tropism. Tissue tropism can be determined by a variety of factors, including local temperature and pH of the tissue, as well as host immunological defenses. The frequency of implantation is greatest where the virus contacts living cells directly, such as in the respiratory tract, in the gastrointestinal tract, in the genital tract, and subcutaneously. These points of contact, where the virus is able to implant itself in live host cells, are referred to as portals of entry. Establishment of infection at the portal of entry may be followed by continued virus multiplication in nearby cells. Spread to adjacent cells also located at the portal of entry may occur extracellularly or intracellularly. At this point, there are two possible general scenarios for pathogenesis. When local sites of implantation are also target organs, and sites of shedding in many infections, the virus does not need to disseminate into the bloodstream in order to reach its target tissue. In this case, localized disease at the portal of entry will occur, and the virus will be shed into the environment as shown in this process to the right. However, in the second scenario, viral spread to cells beyond adjacent cells at the portal of entry may occur through the liquid spaces present within the local site, such as lymphatics or the bloodstream. Dissemination of the virus can also occur by diffusion through surface fluids such as the mucus layer of the respiratory tract. As shown in the diagram to the right, once the virus disseminates, it can reach other target tissues and cause disease. Furthermore, the virus eventually reaches sites of shedding, often called portals of exit, where it can exit the host and be spread into the environment. Now that our discussion of general virology is complete, we will apply this knowledge to one of the largest contemporary issues in modern-day virology: the recent emergence of severe acute respiratory syndrome coronavirus 2, labeled as SARS-CoV-2, and the onset of the coronavirus disease, or COVID-19, global pandemic. Coronaviruses, often abbreviated as CoVs, are enveloped RNA viruses that have become a major public health concern since the SARS-CoV outbreak in 2002. On the left, a thin-section electron micrograph illustrates part of an infected cell, grown in a culture with novel coronavirus SARS-CoV-2 particles being released from the cell's surface. The image on the right was taken from a transmission electron microscope. It also shows SARS-CoV-2 isolated from a patient. Virus particles are emerging from the surface of cells cultured in the lab. As you can see in the picture, the spike proteins on the outer edge of the virus particles give coronaviruses their name, crown-like. The species of origin for COVID-19 has been a major topic for discussion in coronavirus epidemiology. Current research indicates that it is probable that the virus originated in a Chinese bat species. According to a research paper published in the International Journal of Molecular Medicine in 2020, studies in bats have identified viruses originating in this species as the potential vector to human infections. As bats live in colonies, they present a high risk of transmitting the viruses horizontally intraspecies, which contributes to the vertical cross-species spreading ability. Moreover, the research paper states that COVID-19 shares 96% genetic similarity with a coronavirus isolated from Chinese bat species. Additional evidence of bat-to-human indirect transmission of SARS-CoV-2 comes from the fact that two other highly pathogenic strains of coronaviruses, SARS-CoV and MERS-CoV, were identified in both bat and human species and in animals involved in transmission to humans. SARS-CoV-2, like other coronaviruses, is a lipid bilayer enveloped, single-stranded sense-strand RNA virus. Within the lipid bilayer, there are several membrane proteins. Hemagglutinin esterases are a family of viral envelope glycoproteins called receptor-destroying enzymes that mediate reversible attachment to receptors on a host cell. By making the attachment to the host cell receptors reversible, they provide the virus with an escape ticket in the event that it would bind to a false receptor. The M protein is the most abundant membrane protein. Aside from its role in viral assembly, the coronavirus M protein is also believed to have functions in host interactions. The coronavirus E protein is an integral membrane protein. Along with the M protein, E plays an important role in viral assembly. SARS-CoV-2 is approximately 80 to 120 nanometers in diameter, and it contains a nucleocapsid with icosahedral symmetry. Like other coronaviruses, SARS-CoV-2 particles are spherical and have proteins called spikes protruding from their surface. These spikes latch onto human cells and then undergo a structural change that allows the viral membrane to fuse with the cell membrane. The viral genes can then enter the host cell to be copied. The genome of coronaviruses contains genes for the 4 structural proteins: envelope, membrane, nucleocapsid, and spike. Since it is a class 4 virus in the Baltimore classification scheme, it requires a negative RNA replication cycle intermediate that generates protein-coding mRNAs in addition to the genomic plus sense RNA for virus assembly. SARS-CoV-2 spikes bind to receptors on the human cell surface called angiotensin-converting enzyme 2, or ACE2, which is the same receptor used by SARS-CoV. However, SARS-CoV-2 spike protein contains a mutation which makes it 10 to 20 times more likely to bind ACE2 on human cells than the spike on SARS-CoV-1. This augmented tissue tropism provides SARS-CoV-2 with enhanced infectivity, thus allowing it to spread more easily from person to person than other coronaviruses. Furthermore, SARS-CoV-2 appears not only to gain initial entry into target tissue through ACE2, but also to subsequently downregulate ACE2 expression. ACE2 is highly expressed in the respiratory tract, particularly in epithelial cells of the bronchi. Localized pathogenesis of SARS-CoV-2 results in acute lung injury and respiratory distress, as illustrated in the diagram to the left. The viral infection is capable of producing an excessive immune reaction in the host. In some cases, a reaction takes place which as a whole is labeled a cytokine storm. Cytokines are small, secreted proteins that mediate inflammation. Cytokine storms are marked by the uncontrolled elevation of pro-inflammatory cytokines, over-recruitment of immune cells, and systemic inflammation that leads to tissue and organ damage. In the case of COVID-19, the main result is lung inflammation that leads to pulmonary fibrosis or scarring of the lungs, which is illustrated in the diagram to the right. However, ACE2 is also diffusely located on other cells throughout the body. The observation of COVID-19 patient demise on account of heart failure brought about by SARS-CoV-2 infection and the higher risk among patients with previous cardiovascular and hypertensive disease has driven multiple hypotheses regarding potential direct mechanisms of viral action on the circulatory system in addition to its effect on the respiratory system. The treatment for all types of coronaviruses is mainly supportive, as no specific treatment has been discovered to date. Current research is heavily focused on developing vaccinations against SARS-CoV-2 infection. Unfortunately, RNA viruses like SARS-CoV-2 present unique challenges and obstacles for vaccine development. Decades of research has been dedicated to developing vaccines for RNA viruses to no avail. In a report published by the American Society for Microbiology in 2013, researchers state that viruses have high mutation rates and extensive replication capabilities. Viral polymerases can exhibit mutation rates up to 1 in 10,000 base pairs, and some viruses can produce over 1,000 progeny per cell they infect. These high mutation rates and extensive replication capabilities allow for viruses to evolve rapidly. Note here the progression and evolution of increased mortality from SARS-CoV to COVID-19. Just as this novel coronavirus evolved from less virulent strains with lower pathogenicity and lower mortality rates, it is entirely possible that due to the highly mutagenic nature of coronaviruses, which is common across RNA viruses, newer and even more highly infective strains of coronavirus are likely to continue to evolve. However, global research is also ever evolving. Positive actions are being taken, and groundbreaking research is taking place in order to develop serological treatments, create vaccinations, and amplify preventative strategies to slow human-to-human transmission. In this presentation, we have centered our focus on the introductory idea of viruses as simple disease agents that are structurally tailored for infecting host cells and replicating their genetic material. However, as illustrated by the current global pandemic brought on by SARS-CoV-2, there are complex interactions between viruses and their hosts that extend beyond basic virology. These interactions between virus and host influence the world around us and all life on it. Thank you for listening to my presentation. Here are my works cited pages.